Hello, and a very warm welcome to everyone joining us today for our webinar titled Characterization of Protein and Polysorbate Aggregation in Protein Therapeutics. My name is Sophia Hamadash, and I'll be moderating today's presentation. I'm delighted to be joined by our speaker, Khachadourian, Field Application Scientist at Halo Labs, where his work focuses on advancements in biotechnology. This webinar will explore the Aura PTx, a complete protein therapeutic stability solution that enables the characterization of subvisible particle aggregates from developability assessment through to product release. Following the presentation, we will have time for a question and answer session. If you have any questions throughout the webinar, please feel free to submit these in the box to the right of your screen. And without further delay, I'd like to hand over to Dikran for his presentation.
Hello, my name is Khachadourian, and I'm a Field Application Scientist at Halo Labs. Today, I'll be discussing the Aura PTx, a tool that allows for characterization of protein and polysorbate aggregation in protein therapeutics. So why are we discussing particles today? Well, a quick fun fact for you, or maybe not so fun fact, is that particles are the most common cause of product recalls, which is why it's important to not only measure but also characterize the particles that are in your samples. But that still does not tell us why it's so important to characterize particles, or specifically subvisible particles. These are particles that measure on the micrometer scale. Well, these subvisible particles are actually a critical quality attribute. Why? Well, they present safety concerns. They could be immunogenic a lot of times and even cause capillary occlusion. They may result in reduced drug efficacy. And significant particle formation or high particle counts is an indication that your drug has a short shelf life. And for these reasons, the FDA tightly regulates for the number of particles that are allowed in a drug product by following the USP 788 guidelines for particles that are greater than 10 microns in size. And now you may be wondering, how common is it that you come across these subvisible particles in your drug product? And actually, biologics are often plagued with these subvisible particles. And yeah, it could be some foreign contamination or particles from the consumables. However, a lot of times they can be intrinsic, meaning that the drug product itself is highly unstable and is creating these large biological aggregates. And we have two examples of that here. On the left, we have large protein aggregates. They're around 100 microns in size. But on the right, we have a gene therapy product where we have these large AAV aggregates. And we see some smaller ones and some larger ones. But the larger ones are even around 500 microns in size. And my introductory slides might have already given this away, but fundamentally, the Aura serves the purpose of allowing you to characterize aggregates and particle impurities, and this is done by imaging, sizing, counting, and identifying every single particle that is in your sample, thus allowing you to ensure the quality, stability, and purity of your drug products. The Aura builds off of the USP 788 compendial method to offer high throughput, high sensitivity, subvisible particle analysis and ID. So the process of imaging, counting, and sizing particles is very simple and one that we refer to as backgrounded membrane imaging, or BMI for short. So what does the BMI process look like? Well, it's as simple as 1, 2, 3 really. Step 1 being the backgrounding step. Here essentially we're just capturing an image of each well that we will be using. This process is around 30 seconds. Step 2 is loading our samples onto the membrane and applying a very light vacuum filtration to remove the aqueous fraction. So now all that we're left with are the subvisible and soluble particles laying flat on the membrane. Step 3 is capturing a measurement, where essentially we're just capturing a second image of the membrane, but now we have our subvisible particles. And now you're probably wondering, why is the textured background still there? But don't worry, the software is on our side, and we're going to address this in just a second. Today, I promised you crystal clear images of particles, and I did not say anything about a textured background. But luckily, the algorithm is on our side, and it is going to automatically align the raw background that we've captured along with the raw measurement. It will align the two images and subtract the background. And just like that, all you're left with are your subvisible particles. And now we're ready for analysis. Upon acquisition, you are given 3 levels of information. The first level being your particle-specific analysis, the second level being your sample-wide analysis, and finally, your experiment-wide analysis. So these are counts across all samples and all replicates that you've loaded. So what does the particle analysis data look like? Well, first you're given individual images of each particle that was loaded onto the membrane. And we could take this a step further. For each particle, you are given morphological information, such as the particle size, its shape, and its 3D morphology. For example, here we have a particle that is 57 microns in diameter. Its longest extension is 77 microns, and its shortest extension is 64 microns. So now that we've gained some morphological information about the particles that are in our sample, let's broaden our scope just a bit to understand our sample as a whole. So here we have a particle size distribution showing us how many smaller particles we have, as well as how many larger particles we have. So in less than a minute, the software will generate this data for each individual well or each individual sample. And the example we have here, if we look at the 2 micron size range, we're looking at around 7,500 particles per mil. But then if we look at particles that are much larger, so 30 microns and greater, we have a lot of particles there as well, right? So 440 particles per mil for that size bin. And last but not least, we get experiment-wide analysis. So this is data across 96 samples loaded across 96 wells, measured in, you guessed it, 96 minutes. So here you'll have your different samples, including the number of replicates that you've included for each sample. You'll get some CVs for these samples, depending on whether you have replicates or not, and you get to adjust your size bins to your liking. So now we're looking at particles that are greater than 2 microns, but then we also have size bins for 5, 10, and 25 microns and greater. And although the tools of backgrounded membrane imaging are very powerful in allowing you to characterize the subvisible particles that are in your samples with very little volume and such a short period of time, however, we want to further understand the particles that are in the sample. We want to be able to ID them so we know how to mitigate or minimize their presence. And for that, we have 3 different imaging techniques available on the Aura systems, starting with backgrounded membrane imaging, BMI. BMI uses standard bright field illumination to capture all of the particles that were loaded onto the membrane. BMI is great for particle count, size, and morphology. However, if you are looking to further ID the particles that are in your sample, for that we have 2 extra imaging techniques: side illumination membrane imaging, SIMI, and fluorescence membrane microscopy, FMM. SIMI captures particles that are rigid and have some sort of height, so they protrude out of plane, Typically, this is the case with foreign contaminants such as plastics, glass, and fibers. However, if you are looking to ID the biological particles that are in your sample, for that we have FMM, where we incorporate stains or fluorophores to probe for your biological particles of interest. So let's say we indeed have a sample that is plagued with these particles, and they appear in bright field, but that still doesn't tell us what they are. Are they organic? Is it some sort of foreign contamination? Are they proteinaceous? Well, thankfully, with FMM and side illumination membrane imaging, so SIMI, it does not have to be a guessing game. By implementing the appropriate stain, the combination of FMM and SIMI will show you which particles are positive for that specific fluorophore under that respective channel, and which particles are protruding out of plane and potentially foreign. And in the example we have here, we're looking at a mixture of protein aggregates and plastic particles. And in BMI, these two populations of particles would be indistinguishable, as they are morphologically very similar. However, with the help of FMM and SIMI, you'll start getting a signal under the respective channels, so you get to see what's proteinaceous and what is not, what's protruding out of plane, and what isn't. And I agree with you, the images are mesmerizing and very satisfying to look at. However, the data goes beyond just the pretty images. Here we have some more sample-specific data after successfully staining with the appropriate dye or fluorophore. So now the software quickly generates scatter plots, which allow you to play around with the axes and adjust the different parameters to your liking. So in this case, we're looking at the relationship between the size and that specific fluorescence channel, which is our protein channel. And it quickly lets you sort through different populations of particles. So here we're looking at all of the particles that were successfully stained with dioflavin T and thus proteinaceous, and those that are not. I'm sure it resonates with you when I say that the bioprocess is long and tedious, complicated at times, right? Because you want to measure the stability of your samples at every stage of the pipeline, but it's not always possible at every stage because methods that have existed up until now don't allow for low volume capabilities, because let's say you're at the early stages and you just don't have enough sample material to work with. Or maybe you're even at the later stages and you've addressed every question that could possibly come to mind when talking about the physical stability of your sample. But then you get to the post-market stage and you realize that particles are beginning to reappear. So my question to you is, at what point do you think we should measure particles? And the answer should be everywhere, at every stage of the bioprocess. But if you chose the later stages, I don't blame you, because up until now, legacy instruments only worked in the later stages due to high volume requirements, and they don't work all too well either. This is also where USP 788 compliance becomes critical, but we often forget or misinterpret why USP 788 exists in the first place. We want to ensure the stability of the samples in the early stages to avoid particle reformation down the line. And luckily, we have applications that address every stage of the bioprocess, starting with the cell line development stage all the way to the post-market stage. And we will discuss those today. So in the early stages of the bioprocess, so the cell line development stage, you're probably looking to identify which cell line candidate is going to produce the most monoclonal antibodies. What most fail to note is that you also want a cell line that is going to produce the most stable antibodies. However, the cell line that is producing the most MABs is not necessarily producing the most stable MABs. And at this stage, you're limited on volume, so it's very difficult to assess the stability of the different cell lines. But here we've developed an application where with as little as 10 microliters of sample, you could do exactly that. So here on the left, we have an unstable cell line, so unstable map production. On the right, we have stable map production. We've tagged our human IgG with the secondary antibody, and we've stained our CHO cells with dioflavin T. And as you can see on the left, we have far more protein aggregates when compared to on the image to the right, where we have stable map production. So now you enter the developability stage, and you want to assess which molecules are the most manufacturable and which ones are not. So in the example we have here, we have 3 different proteins exposed to 5 different buffer solutions. And so we want to assess their stability in terms of particle counts, right? So we're looking at counts per mil greater than 2 microns. And right off the bat, you see that protein 2 did not enjoy any of these buffers. We see a lot of particle counts across all across 5 of the buffers that we see here. And then so protein 1 and protein 3 were off to the races. And as you can see, protein 1 was very stable in all 5 of the buffers. Protein 3 did okay, but still not as great as protein 1. So with low volume and high throughput, you're able to quickly assess the developability of multiple molecules that you would like to push over to the formulation stage. When you enter the formulation stage, you want to be able to measure polydispersed samples with high precision. And to demonstrate how the Aura does a very good job of doing exactly that, we have 3 different samples. The first one being ethylene tetrafluoroethylene. This is a reference material from NIST. It is supposed to mimic large protein aggregates, a polydispersed sample of protein aggregates. And then next we have stir-stressed human IgG. And then we also have a combination of IgG and ETFE. And not only do we see that the combination of IgG and ETFE totals out at exactly 190,000 particles, which is what you would expect when adding these 2 samples. So not only are we demonstrating high precision there, but then you also have very tight CVs when measuring a polydispersed sample across multiple replicates. Or perhaps you want to understand the behavior of your product. You want to see if your product is what's aggregating, or perhaps there is something else going on. Maybe the particles are foreign contaminants of some sort. Well, this is where the fluorescence membrane microscopy really helps us out. As I've already mentioned, ETFE and protein aggregates are very similar in their morphology. But then when we stain, this is the same sample that we just saw. It's a combination of ETFE and human IgG. When we stain the sample with dioflavin T, you start seeing how many particles are indeed proteinaceous and giving us a signal for that dioflavin T channel. And these are the particles along the y-axis, and how many of which are non-protein, and so those are the ETFE particles along the x-axis. And we also see that the dioflavin T is staining our protein aggregates with high specificity. So even in a sample comprised of different types of particles, you are able to accurately and reliably ID your biological aggregates within your sample to see what is truly going on. And now we are at the manufacturing and release stages, and this is where USB 788 compliance becomes critical. So we have to ensure that our technique is satisfying the USB requirements and of course exceeding. So what are the key requirements for membrane microscopy? in order to satisfy USP 788, well, first we have to make sure that we are scanning the whole analytical area. We also have to make sure that real particles greater than 10 microns are tabulated with differentiation of particles at 10 to 24 microns and 25 plus. Of course, there's another size bin when looking at the 788 or 789 size ranges, that is 15 microns and greater. And we have to be able to validate the technique to ensure appropriate counts and size. And of course, the Aura satisfies all of these key requirements. And now let's discuss how. Backgrounded membrane imaging has always been very powerful in allowing you to accurately characterize the subvisible particles within your sample. And this all started with the Horizon system. And although very powerful, the Horizon would only measure 64% of the well, and it would scale that information up to the full 100%. However, with the Aura systems, we are measuring the full 100% of the well. So we have 100% sampling efficiency. So we have no missing data and no scaling factors. You could accurately count USB standards. Membrane particle distributions can be monitored. And of course, as I just mentioned, USB requires that you measure the entire analytical area, and that becomes very possible with the Aura. And so, of course, part of validating the instrument for USB is being able to accurately count and size. So let's start with sizing particles or USP count standards. Both the Aura and the Horizon have always done a very good job of sizing particles. The example we have here, we have 3 micron, 5 micron, and 15 micron count cal beads, and across 3 different Auras and the Horizon system, you can see that these systems accurately and reliably measured the size of the different beads. Where we start seeing a major difference between the Horizon and the Aura systems is when it comes to validating the instrument with accurate particle counting. And so here we have, again, the same 3 micron, 5 micron, and the 15 micron bead standards. For the 3 microns, you are supposed to measure within 10% of 48,000 particles per mil. And you see that the Aura is right on the money with that. The Horizon slightly overcounts. For the 5 and 15 micron beads, you are supposed to measure within 10% of 3,000 particles per mil. And we are right on the money with the Aura systems. The Horizon slightly undercounts. So with this new development of the Aura, you're able to both count and size accurately and reliably. But now let's fast forward to the later stages of the bioprocess. So this is the post-market stage. And by now you've already established which candidates are the most stable. You've perfected your formulation and manufacturing process. And you've added polysorbate as a surfactant to improve the stability of your product and avoid protein aggregation. But then you start noticing that particles are reappearing. So you're wondering, what went wrong? And actually, the polysorbate itself can go wrong, and it can degrade and form free fatty acid particles. So here I have the 2 main types of polysorbate, PS20 and PS80, along with their degradation pathways. And as you can see, with PS20, whether it's through hydrolysis or oxidation, forms these long-chain fatty acids, more specifically lauric acid, myristic acid, and palmitic acid. And PS80 degradants are mainly comprised of oleic acid, and we do see some palmitic acid in there as well. And because of the hydrophobic nature of these long carbon chains on the different species of fatty acids that I just mentioned, they do self-assemble and form these micelles with a hydrophobic core. At Halo, we like to refer to these as cornflake particles. As you can see in the bright field image, they do resemble cornflakes until we stain them. And we could actually use that micellar structure to our advantage by staining with Bodipy C16, compatible with the second channel on the Aura PTx. And what Bodipy likes to do is insert itself right in the hydrophobic core of these free fatty acid aggregates and give us a strong fluorescence, allowing us to ID our polysorbate degradants in a sample. And now that we've established that we could ID both protein aggregates as well as polysorbate degradants with the Aura PTx, what happens when you have a combination of the two, right? In a realistic situation, you're going to have degradants, polysorbate degradants, as well as protein aggregates. Well, you could actually utilize the two channels on the Aura PTx and do a serial staining of the sample all within the same workflow to specifically ID the two species of particles. So here we have a sample of human IgG mixed with free fatty acid particles. We've incorporated both the Bodipy stain as well as the thioflavin T stain simultaneously, acquired the fluorescence, and as you can see, we get a signal under each of the respective channels, and then we could actually use that scatter plot that we saw previously and plot the protein channel along the x and the free fatty acid channel along the y-axis and get two separate subpopulations of particles. Your free fatty acids along the y and your protein particles along the x. And now to summarize why the Aura PTx, well, some of the many reasons include assessing the stability of your protein therapeutic all the way from cell line development through product release and even beyond, as we saw with the polysorbate application. You get to measure and ID 100% of your particles, so whether it is a protein aggregate or a free fatty acid particle, or maybe even an extrinsic contaminant of some sort. The Aura allows you to measure and ID all of those particles. It is compatible with USB 788. You get to save time, a lot of time actually. So it is a high throughput method. So instead of spending months running your samples, it takes just a few hours. And there is a 21 CFR Part 11 version of the software available. And finally, as you saw, it is very easy to use. And I want to thank you for joining us today. I hope you enjoyed. Now I'll open the floor to any questions.
Yes, thank you for that presentation, Dikran. Like you said, we're now doing a question and answer session. So if anyone has any further questions, please do send those in. And you can submit those by typing them into the question box to the right of your screen. We have had some questions already, so I will just begin. The first one is, since protein aggregates can exist in cell therapy products as well, have you done any work to distinguish between cells and proteins?
Yeah, thank you for having me, Sophia, and Select Science. It's truly been a pleasure. So cell therapy obviously is an emerging field. So in true Halo Labs fashion, we want to remain up to date and flexible with our applications. And as you noticed, our instruments come with different channels compatible with different dyes. And the cell therapy applications are actually very interesting because you could utilize the thioflavin T again to stain for your protein aggregates. And on different models of the Aura, we do have a second channel that is compatible with DAPI and Host. And by utilizing both of the stains, as we did with the polysorbate application, you could actually distinguish between protein aggregates and cells. And that's not just singlets of cells, but if you have doublets, triplets, even aggregates of cells, you will be able to pick up on those.
Great, thank you. So moving on to the next one. We have, okay, so this question is, what if my samples contain silicone oil?
So yeah, very good question, and that comes up. And because we are removing the aqueous fraction from the sample, silicone oil tends to flow right through the membrane. So you will not be measuring silicone oil in the sample on the Aura.
Right. What are the particle size limitations of the Aura?
Yeah, another very good question. I'm sorry if I didn't mention this during the talk. That's probably what I should have started off with, but we are measuring subvisible particles. If we are looking to just count size and gather morphological information with the BMI, the size limitations is a minimum of 2 microns. However, if we want to incorporate a stain or the fluorescence applications, that is 5 microns plus. And the maximum size is actually the size of an entire well, so that is close to 5 millimeters in size. So if you have a particle that is 5 millimeters in size, you will be able to capture it on the Aura.
Amazing. Next one. Do you have membrane options for studies with smaller sample sizes, or are there only 96 well plate options?
So we only have a 96 well option. If you are looking to load lower volume, you could go as low as 5 microliters on these same wells. So you don't have to cover the entire well. You could still get away with using lower volume because the Aura is capturing the entire well regardless.
Great. I think this is our final question, unless anyone wants to send any more in. But this is, you mentioned theoflavin T staining for protein IDs. Would it be possible to use an antibody labeling approach on the Aura PTx?
Absolutely, yeah. So because you are given 2 channels on the Aura PTx, the first channel is compatible with thioflavin T, but you could actually utilize that second channel that we mentioned for the polysorbate degradants. You could actually, that is a GFP channel. So if you would like to incorporate a secondary antibody approach to probe for other proteins of interest, that is definitely an option. And that's actually what we used in the cell line development application. And all of these applications are available on halolabs.com. So if you want to go look at the technical library, we do go into detail with what channels we use, what stains, what antibodies.
Amazing. Yeah, so those were all our questions. Thank you so much for your presentation and for answering all of those for us. If anyone did want to submit any more questions, you can email them over to me, and that would be at editor@selectscience.net, and I will follow up and send you the answers. And also remember that you can download any related resources in the tab to the left of your screen, including a certificate of attendance. And if you'd like to listen to today's webinar again or invite a friend to listen, this will be available on demand in a few days. So goodbye, everyone. And thank you again, Dikran, for the presentation and for everyone joining.
Thank you, everyone.
Bye-bye. Thank you.